Righty, hello everybody. Welcome to another edition of Simon Says. Today I'm going to be continuing with our series on the elements of creation, looking at another non-element, this time protein. Very important in the biology world. Very complex protein, and it's behind more things than you may imagine. So let's dive right in. And so, what do you think comes to mind when you think protein? Maybe it's these things, lots of foods, very important nutrients. You've seen it on your food labels. We know it's important. Our bodies need it. Lots of nice, healthy foods there. Or maybe think protein, you're thinking this guy in the gym building muscle mass. Got your protein shakes, it's to fuel that muscle building. So there's a number of different things that protein can present itself as. But when I think protein, I think this. That's protein. That's beautiful. And we'll talk about that later. But as you can see, there are a number of different ways to kind of consider and ponder protein. So we're going to have a look at the different categories of proteins today, and obviously some examples of proteins and how they fit in there as well. And then as after that, we'll look at protein structure and how that structure helps them to do the things that they do. And then we're going to have a look at when proteins go bad at the end as well. And so you're most likely familiar with at least some types of proteins. So for example, there's insulin. So that's a protein that regulates the level of sugar in the blood. And you may have heard of antibodies, part of our immune system. So they're like a little Y shape and they stick to pathogens, bad nasties in our body, so that they can be neutralized and the body deals with them. There's some others too. There's hormones, so a wide variety of types. You may have heard of that generally. And so their job is to send signals around our body to tell our body to change its behavior. You may also heard of collagen. Collagen's a structural protein, gives firmness to your tissues, helps to keep your face nice and supple. And when you don't have collagen working as a result of scurvy, your body starts to fall to bits. That's not good. You lose your teeth. And then there are enzymes. These are catalysts that help us to speed up chemical reactions. You might have heard of them in washing detergents and that sort of thing, helping to break down bits and nasties that we're trying to clean. And then something even like spike proteins. So on COVID, it has these spike proteins which allow it to bind to human cells. So the COVID virus can actually stick to a human cell, and that allows it to get in and access it and do the nasty things it does to our body. So a wide range of different proteins you've probably heard of in at least one way or another, and there's likely some other ones you're familiar with too. But as you can see, a wide range of things that they do. And so basically, we can think of proteins as being the employees of the body. They have a very specific set of skills and they have a specific job in line with those skills. So they do that job, they do that thing. They're experts, they're specialists. And with a whole suite of all different proteins, about 100,000 or more in a human, for example, different types, all working together, they allow us to have the wonderfully complex bodies that we have that work most of the time. All right, so as you may have noticed, there are different categories of proteins. I think I mentioned roughly one from each before. So we've got those immune proteins, like those antibodies and spike proteins. So they're to do with fighting nasties away, keeping us safe. There's the structural ones like collagen and laminin, helping to hold us together. There's also genetic proteins like transcription factors and repressors. And so they help to essentially turn genes on or off, or to increase them or decrease them in their amount in terms of how active they are in the body. There's transport proteins, so they help to do things like move around sodium and potassium ions. There's literally pumps to move those little chemicals and minerals around. Then there's catalytic enzymes, which help to do chemical reactions, and signal transduction. So getting signals around our body to tell our body to do different things. And so here's an example of a structural protein, laminin. I appreciate this wasn't fuzzy, but this is sorry, it is fuzzy, but this is the picture that I wanted to use because it's this interesting cross-shaped protein. And interestingly enough, it helps to hold our tissues together. It's got these collagen binding domains. And so along with collagen, which also helps us to hold together, it sticks to collagen and then collagen sticks to laminin, laminin sticks to collagens. And it sticks to heparins as well down the bottom as well. So as they all stick together, they form like this it's almost like a big series of band-aids layered on top of each other, causing just sticking together, holding us in place and giving our cells a safe place to attach to and do their job as well. And so laminins, it's a cross-shaped protein that holds us together. And interestingly, in Colossians 1, it says that Jesus essentially holds everything together. And so I find there's a really cool parallel between what that says and a cross-shaped protein that does that job in our bodies. So that's one example of a structural protein, and that's a real microscope image of one there, and compared to an artist's rendition of its different parts, and Jesus superimposed on top, of course. And then we have lysozyme, sorry. So this is an immune enzyme. And so it has a very specific job of hydrolyzing the 1,4-beta linkages between N-acetylmuramic acid and N-acetyl-D-glucosamine residues in a peptidoglycan. Okay, so what does that mean? So peptidoglycan is a large portion of a gram-positive bacterial cell wall. And so basically, it can use water to break bonds between peptidoglycan. And because that makes up the cell wall in bacteria, it can use water to bust bacteria open and kill them. And so that's really cool. So it acts as part of our immune system in order to do that. That's really helpful being able to kill bacteria. And so this is a picture of the protein here. So there's lots of different ways to kind of model and show proteins. And so this is what we call a space filling model. It's very blobby and shows the physical space that it would take up in 3D. And we've got these colored sites here. So the blue is the binding site. And so this is where the water and the peptidoglycan come together. And so the lysozyme attaches them and it uses its very specific shape and chemistry to hold it. And then at the red part, the catalytic side, that's where it brings them together. It's like this matchmaker wrangles them and boom, chemical reaction happens when they come together using that water to break the bonds. And so this is a reaction that may or may not happen naturally. And if it does, it's very, very slowly. But an enzyme like this, its job as a matchmaker to bring things together and force a chemical reaction can cause those reactions to happen 1,000 or 1,000,000 times or even faster. And so it allows bodies to do chemical reactions that are important and do them at a rate which is carefully controlled, so our bodies can do the things that they need to do. In this case, killing bacteria to keep us safe. And then another example is MAP kinase kinase kinase. And I love MAP kinase 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 because its name is ridiculous. And so it's part of a signal transduction pathway. And so its job is to move signals through our bodies to do stuff. And so on this image here, I'm not going to talk about that, but basically signals are very complicated. This signal, there's a messenger that tells this messenger, which tells that messenger, which tells those two messages, but not that one. And then that one tells that one. And so we get these really complicated pathways, and scientists dedicate their entire careers to looking at one little particular signaling pathway. So I mentioned before, our body's got over 100,000 proteins, and a great many of them are these signaling proteins, which act in combinations and chains in order to tell our body to do the right thing at the right place at the right time. And so this protein has a ridiculous name because it's to do with kinase phosphorylation. So a kinase is the name given to a protein that phosphorylates another protein. And so phosphorylating is basically sticking a phosphate group on it. And when you stick a phosphate group on a protein, it can turn it on or off like a switch, or it can mark it to be destroyed or to be stuck in a certain space. So it can be like a label or a switch to turn it on and off. And so obviously this helps us to manage proteins and what they do and their action and where they are. And that helps the body to do what it needs to do. And MAP kinase 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 means it's part of a chain of pathways. And so MAP kinase 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 phosphorylates and adds that molecule to MAP kinase kinase. And then MAP kinase kinase phosphorylates MAP kinase, and then MAP kinase phosphorylates MAP. So the MAP can do its thing. And what's MAP? Mitogen activation protein. It responds to osmotic shock or heat or inflammation, a range of particular things in the body. The body's like, oh, hang on, I need to get onto this and do something about it right now. And so the reason we have these complex chains of signaling is because it allows the body to ensure that the signal gets there and in the appropriate amount. And so we start with one little receptor, one little signal. It tells one messenger. That's fine, but are we going to trust one messenger? But that messenger will phosphorylate a series of other messengers. And then those series of messengers will phosphorylate multiple other messengers. And suddenly you have this army of messages sending this massive signal, this giant swell, and the cell's like, right, I've got to do something about this right now in a big way. And so that's the benefit of these long signaling pathways in these chains, using those phosphorus groups to mark things or activate them or turn them off. And so a lot of our cells signaling proteins operate in a similar manner. And so you may be asking yourself, how in the world does this happen? These things don't have brains. They're little chemicals, they're little tiny molecules. There are thousands, squillions of them running around in each and every one of your 70 trillion cells. And they don't have brains, but yet they still have a specific set of skills and a specific job that they carry out in line with those skills. They do this without thought. They don't have brains. They don't communicate with each other apart from, phosphorylation, little things like that. They don't talk. They don't have apprenticeships or university degrees or instructions. How do they do this? The answer, of course, is chemistry. And so chemistry is all about how little chemicals come together and interact in certain ways. And those interactions dictate the things that result. And thankfully, because God has custom designed us, that they all work together in a way that allows us to function. And so a really important aspect of this is the structure of a protein. So we're going to look at the different aspects of structure. So there's the primary, which is a sequence of amino acids. You might have heard of that before. They're like the little Lego blocks that make up a protein. Then there's a secondary structure where those acids arrange themselves in specific structures. Then we have the tertiary structure, which is the overall protein that may result. And then quaternary, where multiple protein strings can come together in a more complicated, larger way as well. So we're going to go through each of these to show the importance of the chemistry and how the proteins actually do the things that they do. All right, so our first structure is the primary structure with our amino acids. And so an amino acid is the name we give to a compound which has an amino group and an acid group, acid carboxyl group, and a unique side chain. So there's a few examples here, lysine, glutamine, and aspartate. And so each one of them has an amino group, this NH2 on the side. They each have this acid carboxyl group, the CO and the OH, that's our acid group. And then they each have this unique side chain in the blue here. And so this unique side chain is what gives them their name. That's what makes them special and unique. But amino acids are really interesting because they can form really long chains. So these carbons and these nitrogens connect together. So we can have this huge series of amino acids all connected in a big long rope. And that rope has these unique side chains hanging off the side with lots of different bits of chemistry resulting. And so when we have amino acids coming together in a string, we call that a peptide chain. And so you may have heard of peptides. They're just a little string of amino acids. And they come together with what we call the peptide bond. So that looks like this one. So we've got two alanine amino acids, a nice simple one. We've got that amino group and the acid group here. And as those two come together, we release some water, H2O, and they come together to form this peptide bond, forming a nice strong bond. And as many amino acids come together, we get the chain that looks like this. So we call this our primary protein structure. And so all of these colors represent different amino acids. We have 20 different amino acids that we use in the body, and so 20 different colors. They can come together in all sorts of shapes. Well, not shapes, well, they each have their own shape, but they can all come together in combinations as they form this big rope string, and they're all connected by that amino connection group. And so these are all the 20 amino acids that are commonly used in the human body. And so scientists basically assign them a letter of the alphabet. So we've got 20 letters in the alphabet, that's most of them. Each one of them relates to an amino acid. And so when scientists talk amino acids, it's a string of random letters where each letter is referring to a specific amino acid. And they've also got little abbreviations too, but typically you'll see the letters. And so this crazy thing is a table which illustrates how DNA encodes for specific amino acids. And so the reason that DNA in our genetic code is so important is because it tells our bodies the ingredients, the instructions to make these proteins to do stuff. And so you are you because of your unique blend of proteins, which made you short, tall, good with your hands or better in a classroom, those sorts of things. And so our genes dictate that by the type of proteins that they tell our body to make. And so these letters are the DNA code, A, T, C, and G. But when we make proteins, the T's become U's. It's the simple thing to that. But basically, we read these A, T's, C's, and G's, your DNA code, which is just a 3 billion long string of A, T's, C's, and G's. And depending on how they come in sequence, it tells us what amino acid to use. So for example, where is methionine? Here we go, AUG. And so if your DNA letters say A followed by a U followed by a G, proteins in your body can read that and go, okay, I need to put a methionine amino acid there. And so we call that our start codon. Our amino acids always start, sorry, our proteins always start with a methionine. And then we might look at the next three letters. Maybe they're CCC here, and then the body knows to add a proline as the next amino acid. So it keeps reading that sequence, keeps adding amino acids, and eventually we custom build a protein that's just right to do the job that we need. And so this is a picture of a little tiny protein factory. Your cells, each and every one of your cells has many of these factories inside it. And so at the bottom, we have this string of messenger RNA, which is basically a short-term copy of your DNA. And it'll read 3 of these A, T, C's, and G's at a time. And each of those 3 will cause a tRNA, this molecule here, to come in and bring the exact right type of amino acid. So these specific codes will bring that specific type of amino acid, and then we build that protein chain custom built from the code to design. And so it's interesting to note that the DNA code is the only code known to man that was not invented by man. Every code, whether it's language, music, Morse code, they're all codes invented by humans. DNA is the only code that was not invented by humans. And that's interesting because a code implies meaning, and meaning implies intent, purpose, and intelligence. And so that's a really cool evidence that I believe that we were made because there's a code built into us that we need to function and survive. And intelligence is the only way that I know how to explain a code. All right, so next we have secondary structure. Don't worry, that was the big one. These are a bit more straightforward. So secondary structure is what happens after we've got that nice long rope of amino acids. They then start to arrange themselves in particular shapes. So you can see I've got 3 different amino acids here. I'm labeled, and then the rest just continue on. But basically, a certain string of amino acids will arrange itself in a nice coil like this, and we call this an alpha helix. Whereas other combinations of amino acids will arrange themselves in this beta pleated sheet. And so they have like this twisted yet straight structure to them. Then these little curly bits that connect them, we just call them random coils. They're just like connectors between the sheets and the coils. And so this is a result of hydrogen bonding, which we've touched on before. And so they're little weak temporary bonds between oxygen and hydrogen, just like we experience with water. And so because they're part of our peptide bonds, when we link a whole bunch of amino acids together, we get those weak interactions, and they allow them to form these specific shapes. And so this is an example of a different way of representing those. So we call this a ribbon model, just because it looks like a bunch of ribbons come together. Whereas this shows the little sticks of the specific molecules. And so all these inside the spiral, there are carbons and nitrogens linked together in that chain. And all these dangly bits hanging off the side are those unique side groups, which give each of those amino acids their unique flavor. All right, so the next one is tertiary structure. We've got a long string of amino acids. Some of them coil up, some of them come in sheets. But this is where those unique side chains come to the fore. And so each one of those side chains has a bunch of unique likes and dislikes. And so we're going to have a string of literally hundreds of these guys, each with their own likes and dislikes. And they're going to arrange themselves spontaneously in a way that makes them happy. They're going to hang out with their friends who like the things that they do, and they're going to stay away from the people who don't like the same things as them. And so these interactions form this big chain to, instead of being a long string, to actually fold and come together in a very specific, unique shape. And it'll come together in that same shape every time, as long as those amino acids are still in that same order and produced to that custom design. And so we have some examples of hydrogen bonds. So we've got hydrogen bond here. So some of the side chains have oxygen and hydrogens. So you get those weak interactions. We get ionic bonds, which is positives and negatives coming together here. So this NH3 group has a positive charge. This O minus has a negative charge. So those opposites attract each other. They want to hang out. There's covalent bonds. And so we have this disulfide linkage down here, sulfur bonds with sulfur in a more permanent, stronger reaction. We call these cysteine bridges as well. So cysteine being an amino acid bonding to another cysteine. And then hydrophobic interactions. Some of these guys just hate being near water. And so they'll hide in a little pile of things that hate water and try and keep all the water on the outside so they're all safe. And so when these interactions come together, they fold and arrange themselves. We get this very specific, unique structure of an amino acid, protein. And so these amino acids they are grouped into their different groups. And so there's a bunch of non-polar ones, a bunch of polar ones, and then there's positively charged ones, negatively charged, and so on. So we can group them by their characteristics as well. When they come together, you get something beautiful like this. And so this is salivary amylase with a couple of chloride and calcium ions. So some little, what do we call them? Minerals in there to support the action too. And so amylase, its job is to break down starch into sugar. And so when you're chewing on something starchy in your saliva, there's this amylase protein, and it helps to break down that starch into sugar. And so this is what it looks like. And so its unique shape allows it to come together with starch, holds it together, and do the chemical reaction that breaks it apart, resulting in sugar. And so all of those amino acids have come together to form these coils, to form these sheets. And then those coils and sheets fold on top of each other, coming together uniquely because of those side chains that result from each amino acid. And then lastly, we have our quaternary structure. And so basically the previous stage was a complete protein, but some proteins are even larger and more complex again, and they're basically made up of smaller proteins. And so this is hemoglobin. And so it has two beta units, so two different green units at the top, and then alpha units at the bottom, the two different yellow ones. And those four subunits come together to form one hemoglobin. And they each have a heme group, which is another molecule in the middle. And when this shape comes together in that way, it's really, really effective at bonding oxygen in a way that allows it to be carried through our bloodstream and delivered to where it needs to be. So not only will the oxygen bind nicely to that, it will also unbind at exactly the right place in the low oxygen area of your body. And so as it moves around, it gets refilled with oxygen at your lungs. And as it hits your peripheral tissues, which are crying out for more oxygen, that oxygen will naturally be released from this shape. And so only some proteins have this quaternary structure. And basically, it's just a pile of other proteins coming together, more connected to form a larger complex structure. All right, so that we've had a look at how the chemistry tells us how our proteins come together. We've seen how they're custom made based on our DNA code. And we can see that how their job results because of their specific shape, which allows them to interact with the specific shapes of other chemicals and to come together with the things to do the job that they need to do. But we live in a fallen world post-sin. Creation was originally very good, but sin has made it not so good. Sometimes things go wrong. So we're going to have a look at what happens when proteins go bad. And so one example of this is phenylketonuria. Tricky word to say. And so you might have been having a look at an artificially sweetened products, and a lot of artificial sweeteners contain phenylalanine. And so you'll see it in your warning label here, right next to contains caffeine. It also contains phenylalanine. And so I thought that was a nasty thing to be worried about until I went to university and found out it's an amino acid. Your body needs this or you die. So why is there a warning label on your Coke bottle that says it contains phenylalanine? And the reason for that is there's a rare genetic disorder, phenylketonuria, or called PKU for short, and it results in a mutation in phenylalanine hydroxylase gene. And so that's the part of the DNA which gives that specific code to make the protein phenylalanine hydroxylase. And its job is to process phenylalanine so that your body can break it down if there's too much, if it's not needed, to make it into something else more useful. And so these people have a mutation in that gene, which means that their body basically can't break down phenylalanine. And so if you introduce phenylalanine into the body, it's just going to build up, build up. So there's a point where it becomes toxic. Your body doesn't have anywhere to put it. It causes problems with chemistry. And if it's not identified and controlled, it can actually cause disability, brain damage, those sorts of things. Because basically you slowly poison the body as you eat regular foods, because just about every food contains protein. And so people with PKU, they need to avoid these artificially sweetened products, which can be particularly high in phenylalanine. Like you don't expect Coke to have a lot of protein in it. But they also need to carefully monitor their overall protein intake, knowing that their protein is going to contain a portion of phenylalanine inside it. And so that's one example of when our body can't deal with a specific amino acid, it causes problems. And there are also prions. Prions are a type of protein which have misfolded. So they've come together in a way when it's done those unique interactions between those side chains. Maybe the wrong amino acid was put in the wrong place and suddenly they don't fold in the right way. Things go different. The protein can't do its job anymore because it doesn't have its shape. Not only that, but it actually does a different job accidentally just by fluke. And it just happens to misfold other proteins of the same type. And so basically, this misfolded protein, through chemistry going wrong, can create copies of itself and make other misfolded dodgy proteins, which make other misfolded dodgy proteins. And so as a result, this can cause cell death. And so we've got normal tissue at the top left there. This might be in a brain, for example, but can also happen in the heart, I believe. But in these damaged tissues, you can actually see holes in the tissue that have resulted from these prions dealing damage and causing cell death, and therefore holes in the tissue, holes in your brain, which is not good, as you can imagine. And so that's scary. But unlike bacteria, viruses, fungus, that sort of stuff, other infectious agents, prions are proteins. They're not living. So if they're not alive, how do you kill them? How do you kill something that is already dead? It's like a zombie protein coming to make holes in your brain. And so this is a really big problem because they're very hard to deal with. And so one example of this is mad cow disease, more scientifically known as bovine spongiform encephalopathy. And so humans can contract the prions that cause this disease by eating affected meat from cows, which contain that prion. And so obviously we need to be really careful to control our farming techniques to ensure that that's not happening, that it's not spreading amongst the cows and then humans to eat them. But what's really difficult is that it can only be detected and seen post-mortem in brain tissue samples. And so when something dies, we need to be thinking that's strange and then have a look at its brain to see if those holes are there. That's the only way we can know that if it's present. So we can only detect it after something is dead. And so that's a really big problem in terms of knowing what's happening and therefore being able to control it. And not only that, when we do that brain tissue and we have a look at it, we have to throw away the scalpel at the end because if there's prions on it, you can't get rid of them really. We need to incinerate it and never use them again. And so big deal, very hard to control once it's happening. And so there are now restrictions on feeding offal to animals. And so some farm animals will be fed the offal from other farm animals. And so that's just a way of spreading the cycle more. So we now have more stringent rules about how they're fed to try and reduce the risk of them picking up the prions. So in turn, humans aren't being affected as well. And then there's also special rules on disposal of abattoirs, particularly with specific parts of the animal which are more at risk to being carrying these prions. All right, guys, so there you had it. We've had a look at protein categories, some different examples, and what they do. Some you may have been familiar with before, but hopefully some new ones as well. We had a look at how the structure of proteins is dictated by their chemistry, and then how that structure dictates the job that they do, and how this was custom designed by God with the DNA code. We've had a look at what happens when proteins go bad as well. So as always, thank you for listening. I'm looking forward to seeing you in the next video as well. In the meantime, I'll see you later.